Welcome back to our class. Today we're going to be looking at meiosis questions. I know that meiosis is one of the topics which is a little bit easy to teach, but when it comes to the answering of questions, it's a little bit difficult. And we know that students are going to be answering questions concerning about meiosis in term 1. So we are preparing you to write the questions concerning about meiosis. By the end of this lesson, everybody must be able to answer questions concerning about meiosis. Since this class is live, if you have any challenge, drop your question. I will be able to answer it immediately so that we can continue together. It's a morning class, I know, but I know that when it's morning, most of the students, the brain is a little bit what? Fresh. So that if it is fresh, it means that you'll be able to grasp. So there are a lot of questions we're going to be dealing with. Some of those questions, you see them on the screen. They will be related to that. But before I go to these questions, let me first give you some few tips when you are going to answer questions concerning about meiosis, where students tend to lose these marks. First of all, you need to know what you call spelling, because this is a very big challenge to students. They tend to ask that, but sir, only a simple just one letter has been missed. Can they mark me wrong? Yes, we will mark you wrong. I always ask you a question that if I spell your name and I miss one letter like this, will you be able will you be happy? Will you be the same person? The answer is no. Then why, when you are writing, you don't write the correct spelling? So spelling is very important. Number two, the things which make students not to get a distinction is when you go about question concerning about meiosis in terms of chromosome number, you see students, they ask them question, how many chromosomes do you see? Students, they think that wherever question comes in meiosis is about human. No, the question can come, but when it is not about human. So don't only think about question that the chromosome number are always 46 chromosomes. No. Sometimes the question will come and then it's not asking about 46 chromosomes. It's asking about the chromosomes you see in the diagram. How many chromosomes do you see in the diagram? Then you'll be able to, let me just give you a simple answer. Look at this. Ne? If you look at this, yes, this is not a question concerning about human cell, because they are not talking about human cell here in this question. No, they're not talking about that. What are they really talking about? They're talking about what you see there. How many chromosomes do you see there? The parent cell, you see that the parent cell had 4 chromosomes. Then why do you think about 46 chromosomes? Please try to not to talk about 46 chromosomes whenever they ask you a question concerning about meiosis. All right, let's start with these questions, and then we see how we can answer these questions. I will make these terminologies the end. Yes, I will also come back to them and then go through them. And then I'll also relate you to our booklet of the distinction material. Yes, this book, you know it. Always find the lessons here. You'll find them there. So we are going to be looking at this. Okay, the first question is asking. They're asking, what are they asking about? Let me know if you have any question when we are going through this. If you have any question, let me know so that at least I help you out. All right. The diagram below represent different phases of meiosis in the diagram. So if you are looking at this, what are these phases? To differentiate between meiosis 1 and meiosis 2, how do I differentiate this? The difference is 1. What is that difference? The difference, if this is a chromosome, a cell, and then I have a chromosome, I have 2 chromosomes there. So if they ask you, how many chromosomes do you see here? Don't say 46 because you don't see 46 there. I see chromosome 1, chromosome 2, chromosome 3, chromosome 4. So I see 4 chromosomes. So how do I know that this is meiosis 1, this is meiosis 2? I will only know if these chromosomes are in pairs. If they are in pairs, yes, you see that they are in pairs, therefore it is meiosis 1. They have not yet divided. If I see that these chromosomes are single, like, no, they are single like this. For example, I will know that this is meiosis 2 because the chromosomes have already divided. They divide. When they divide like this, so this one goes this side, they form a cell. So you see that now this one is this. So when the chromosomes are in pairs, then definitely we shall say that that chromosome is under meiosis 1. If the chromosomes are single, then we shall say that that chromosome, that cell is under meiosis 2. So now look at this. Are they in pairs? No, they're not in pairs. You see, they're not, it's single. This is longer than this, therefore they can't be homologous chromosomes. What about this? Yes, these are in pairs. You see, they're in pairs. If they're in pairs, what does it mean? It means that this phase is meiosis 1. What about this? They are single. You see, they are single. Therefore, that process or that phase is meiosis 2. That is the criteria you must use to know that this is meiosis 1 or it is meiosis 2. Number 2, to know which phase is it. Yeah, because now I'm just going through highlights so that when I push down, it will be a little bit faster. So, number 2, these spindle fibers and the centrioles, they are supposed to be in your sides. Let me show you what I'm saying so that you know exactly which phase is it, because I can ask you, which phase is this? Sometimes we bring it like this, sometimes we change it and the faces are like this. You see, now the spindles are facing this side, but if you look at this, the spindles are facing the other way around. So try to change it, ne? So that now, yeah, they are supposed to be inside. Now you see, now the chromosomes are in the middle. Therefore, you know that now this is what? This is the metaphase. But because they are in pairs, remember I said that if they are in pairs, therefore it is meiosis 1. Therefore, it is metaphase 1. That's how you can differentiate them. Yeah, so when the question comes in and then the chromosome, you see, now these spindles are up and down. No, they're supposed to be inside. So you try to rotate your paper until you see that now, yes, you see now the chromosomes are inside. That's how you can identify whether it's meiosis 1 or it is meiosis 2. All right. Now let's start answering question. Identify what is A. A is the center of the chromosome. Therefore, it becomes centromere. Centromere. What is B? They're saying B. What is B? B. I talked about this. What is B yesterday? Ne? What is B? Chromosomes which have the same shape, same size, same location of genes, not same genes, same location, same locus. Yes, we call them homologous chromosomes. Guys, don't forget to write correct spellings. Homologous. Yes, homologous chromosomes. So you have to know that this is the homologous chromosomes. What is C? spindle fibers. We accept spindle fibers. We also accept spindle threads. Yes. What about, they're saying that, so we have tried to name everything which they need. But what about this? How do we call this? What is this? So this is what? How do you call this? This is cell membrane. It is not a nuclear membrane. It's a cell membrane. It's a cell membrane. Yes. So, identify the phase represented in diagram 3. So when you come here in diagram 3, I say that now these are supposed to be inside. So try to rotate it with your paper, with your paper, and you see now they're inside. You see the space? It means that the chromosomes are apart. Because they are single, therefore is meiosis 2. Because they are apart means, it means anaphase. Therefore, the phase is going to be anaphase 2. So the answer becomes anaphase 2. Identify the phase represented. We have identified that. Write down the number of diagrams shown in sequence in which the phases occur. So where is the number? Okay, wow. First identify which one is meiosis 1 and which one is meiosis 2. So we see that, okay, this is meiosis 1. Yes, this is meiosis 2 and this is meiosis 2. Yes, that's the first step. So it means that definitely diagram 2 is going to be the first one. Now what about between this and this, diagram 1 and diagram 3? Which one is going to be meiosis 1? Which one is going to be the first? Which is going to be the last? This is prophase. Yes. And then this is anaphase. Therefore, prophase comes before anaphase. Therefore, it's going to be diagram 1 and then it's going to be diagram 3. Guys, let me know the questions you are asking. If you are not clear on what I'm saying, please let me know. I'll answer your question immediately. Yes. So I'm waiting for your comment if you don't get something right, or you have a challenge I've not talked about when I'm trying to discuss about it, so that by the time you write your end of term, you are able to answer these questions. Yes. Then they're saying that state one difference between metaphase 1, metaphase 2, and metaphase 2. Basically, this is supposed to be metaphase 1 and metaphase 2. Metaphase 1 and metaphase 2. Yes, metaphase 1 and metaphase 2. So what is the difference between metaphase 1? Remember that in metaphase, chromosomes are in the center. You have a cell, Yes, you have chromosome. Yes, chromosomes. They are supposed to be in the center. Therefore, that is the commonality both of them have. But in metaphase 2, because the chromosomes have been divided, have been split, the cells have split. Yes, cytokinesis has occurred. Here in metaphase 1, chromosomes align along the equator in homologous pairs randomly. And then here, chromosomes align along the equator singly. Look at this book. What we said here nicely is stipulated there what you're supposed to talk about when you come. Homologous chromosomes randomly align along the equator in 2 rows. And then they're saying each chromosome is attached to the spindle fiber. What are they looking at? Here, homologous chromosome. This is the key point they are looking at in this question. If they say difference between meiosis 1 and metaphase 1 and metaphase 2. In metaphase 1, homologous chromosomes align along the equator. You see? Yes. In pairs, you see? Yes. While in meiosis 2 or in metaphase 2, what happens? Yes. What happens in metaphase 2? Check, they are individual, not homologous. So that is the major difference what you're looking at. So chromosomes in metaphase 2 align along the equator singly or individually, while here chromosomes align along the equator in homologous pairs or in pairs. Yes. So that is the major difference what you are supposed to talk about. If that question comes, we like to ask metaphase. So you can't go to the paper without knowing the concept of metaphase. You can't go to the paper without knowing the concept of prophase, whereby crossing over is taking place. Yes. After going through this, you won't regret when you're going to write the paper. Before even you write the paper, you have to come back and then watch this video so that, yes, your time for revision is minimized. Okay, the diagram below represents one cell in the phase of meiosis. So look, they are not talking about this is a human cell. Please don't think about 46 chromosomes. I repeat, don't. So they're saying that identify the phase of meiosis shown in the diagram. What is it? If I look at this, let's first label and then you find out. What is A? A is a cell membrane. You can't say nuclear membrane because the nuclear membrane, you can only see it during interphase and prophase. And towards the end of prophase, the nuclear membrane is also disappearing. You also see it in the late telophase, but we don't talk about here late telophase, early telophase. We don't talk about it. Yes. So you see it, you can see it at the end of meiosis. So, okay, this is a cell membrane, and then spindle fibers. I said sometimes you call it spindle fibers, sometimes you call it spindle threads. Identify the phase. So, since now the spindles are in the sides, therefore this is in the center. You see, chromosomes, they're in the center. Therefore, the first thing you think about is the metaphase. Metaphase. Yeah, but is it metaphase 1 or is it metaphase 2? Because the chromosomes are single, you see, single. There is no chromosome here. There is no chromosome here. Therefore, it is metaphase 2. Therefore, it is metaphase. So the answer becomes 2. Metaphase 2. Yes. So, I think I'm clear. There. Just a small thing here. Trying to, yeah, good. Yeah, perfect. All right. They're saying that give one function, give one observable reason for your answer. Why do you say this is metaphase 2? Chromosomes align along the equator. That classifies to be metaphase. Chromosomes align along the equator singly. Singly classifies it to be 2. Metaphase. Along the equator, chromosomes align along the equator. That makes it to be metaphase. Why do you say 2? Singly. They align along the equator singly or individually. So it classifies it to be metaphase 2. Look at into your our booklet. Individual chromosomes line up at the equator of each cell. So individual chromosome makes it to be metaphase 2, that is meiosis 2, and then align along the equator. This makes it to be metaphase because meta means middle. Yes. So when you are giving the reasons, you're supposed to don't just say chromosomes at the equator. Which kind of chromosomes? Yes, it means that what you have defined there is you are just classifying it to be metaphase. But is it metaphase 1 or it is metaphase 2? That's how students tend to lose the marks. So it is metaphase 2. So they align along the equator in singly. They're saying that identify structure A and B. We identify them. A, we say the cell membrane. B is the spindle fibers or spindle threads. And then chromosome 2. This is a chromosome, Yes, a single chromosome. Describe the role of B in the movement of chromosome during meiosis. What is B? Spindle fibers. What is the function of the spindle fibers? Yes, it pulls the chromosome or chromatids, yes, to opposite poles. Yes. So we say that this centria attached to the spindle fiber. So when these centrioles they move, né? They move, they split, they tend to pull these chromosomes. You see that they are being pulled. What pulls them? What pulls them is the spindle fibers. So the spindle fibers pull these chromosomes or chromatids to opposite poles. And also, if they're saying that these spindle fibers, they attach themselves on the centromere, and then during the separation of these chromosomes, then they are pulled to opposite poles. Then they're saying that draw a labeled diagram of structure C. Where is structure C? Yes, of this chromosome, yes, as it would appear in the final phase of this meiotic division. Show the correct shading. I want to talk about it. So it's 4 marks. And they're saying that they want this chromosome, how it would appear. So if you're looking at this chromosome, make sure that you draw with the correct shading. If you take this chromatid, it means that it must have this. If you take this chromatid, for example, if you take this chromatid, then it means that it's supposed to have that. While if you take this chromatid, it's supposed to have that. But they're saying you're supposed to draw it, a labeled diagram. How will it appear? Yes. Remember when this is happening, also this will happen. For example, this can go with this. So when this cell divides, so you have to either you take this and this, or you take this and this. The shadings must be there. So if you take this and this, then automatically you're going to have, for example, let me give you a cell, and then final stage. Final stage, it means that it is telophase. So telophase 2, the chromosomes are supposed to be singly. In telophase 2, the nuclear membrane is there. The centrioles are there. This chromosome, if you say this chromosome, if I take this side, it's not shaded. What about if on this one, if I take this side, yes, whatever you take, make sure that it is the correct shading. And then one part on top is shaded. Not good in drawing. Okay, let me draw it. There is no person who is a scientist and is not good in drawing. Yeah. All right. They're saying that. Okay. Let me draw it here. Yeah. Okay. This is shaded and also this is shaded. Yeah. You see? Because this part is shaded and this part. Those are the things where you lose marks. Because just shading is a mark. They don't stop here. They're saying that this is 4 marks. You write a title. We need a caption here. Caption. You don't stop there. They say that labeled. You see, it's a key point. Labeled diagram. Ne? So, correct shading. This is a mark. This is a mark. Because this is instruction. Ne? This is instruction and how they will appear. The appearance. Final stage. We need to know that the final stage you have given is telophase. Yes, telophase 2. So it's a mark. Marking, drawing the correct phase is a mark. Drawing the shape of the chromosome single is a mark. That's appearance. And then drawing correct shading, ne? Yes. And the number is a mark, and then the caption. So you see how marks are being split. So if you don't just, if you just draw, you'll end up losing marks. Let's go to another question. Please, if you have any question or I'm rushing, let me know. Yes, I will try to help you out. Ne? Yes. Let me know if you are on online, online, so that we credit you. Yeah. All right. We are saying that diagram 1 to 3, below show some of the phase of meiosis. Some of the phase. It means that it's not the complete. And they're not saying that this is a human cell. I will repeat that. Identify the phase represented in diagram 1. In diagram 1.1. Okay. Diagram this one. And then diagram. Okay. Which phase is this? First of all, the chromosomes are in pairs. So it means that it is meiosis 1. First phase. This one, the chromosomes are still in pair. Also, this is meiosis 1. Yes. What about here? Chromosomes are single because this and this are they're not the same. So this is meiosis 2. So now, you have these two. Which one is first? Because meiosis 1, meiosis 1. So this, I'm trying to show you which one is meiosis 1, which one is meiosis 2. So because the chromosomes are in pairs, chromosomes are in pairs, you see? They're in pairs. And along the equator, you see? So it means that this is metaphase. Metaphase. But you have already said this is meiosis 1, therefore it's going to be metaphase 1. And then because the chromosomes, they are moving apart, so this is anaphase. Anaphase. So automatically, we have identified it. Yes, we have identified it. Thank you, Lawrence. Yeah, we have identified it. So it means that now you have, what do you have here? What do you have? It means that this is anaphase 1, metaphase 1. What about this now? There is a, this could be telophase 1 or prophase 2. But I'm not seeing here centrioles. Therefore, we can classify it as telophase 1. But sometimes the diagram may be a little bit misleading if you don't read the question. Why am I saying that this is a telophase? It's because of the cytokinesis happening. Yes, and this is appearing. Yes, because the late telophase and early prophase, they look almost the same because it is now shifting from telophase to prophase. But we don't talk about late, early. No, we just say telophase, we say prophase. So if you're reading some books, if you want a distinction, just focus on this book, this book, this book. This is what I'm saying. If it is in telophase 1, you see? Yes. They look like this, you see? So now they're supposed to have the, and then you see that it looks like this. Okay, now let's go to the questions. We will find out what is it. Identify the phase represented diagram by 1, I think it's this. So this is the metaphase 1. Identify the phase. This is this. Yeah. So this is gonna be telophase 1. We don't have the spindles yet. Centrioles yet. If the centrioles are there, definitely that becomes prophase. So that's what I'm saying here, that if these would have been there. But in here, we don't have those centrioles yet. Yes. Okay, they are saying that give the letter only of part that, number 1. What happens? What happens? Number one, that contains DNA. DNA, it means that either is a gene, either is a chromosome, a chromatin network, all those things. So in this case, we have chromosomes. So the answer is going to become B, because chromosomes are made up of genes, and the genes they make up are DNA. DNA they make up of. If DNA is coiled with histones, those proteins called histones, they form what called the chromosomes. I explained this before, that chromosomes, gene, chromatin network, nucleotides, those things are almost the same thing, but on different scale. As I can say that you are found in South Africa, in Durban, wherever where you are. It's the same thing, you are in Africa. You understand? Yes. So attached to centromere of the chromosome, attached to the centromere of the chromosome, what is attached? Is the spindle fibers or spindle threads. So the answer is C. Don't forget that they are not looking for the name. No, they are looking for the letter. Only letter. If you give a name, we will cross it, because we didn't ask you. We didn't ask you. Yes. We didn't ask you about the name. So you can't leave such question unanswered, because they are looking for only what? They are looking for only letters. They are not, you don't even need to stress too much. They're saying that name the letter that forms the spindle fibers or spindle threads. What forms are the centrioles? So you come here and look for the centrioles. Where are the centrioles? It's D. So the answer is D. Name the organ in human. You see now, the question now goes to the human. Meaning that all what you have been talking about is not about human. It's just about meiosis. Okay, name the organ in humans where meiosis occur. But they are specific, not just human. They are looking for human male. You see? Yes. We said it here very clearly in our book. We are very clear when you are talking about this. In human or in animals, it occurs in testis, male, meaning that this is the answer, testis. And then in female, it occurs in the ovary. You see, that's the answer. In plants, female, ovary, what do you produce? Ovules. And then in male plants is anthers, what do you produce? Is the pollen grain. So you see, download this book from this by join the group and get all the books. Yeah, so you can download it. I'll send you this. I'll put it after the lesson. I'll put it in the description below, so that you have it, and then you can click on that link so that it will take you, and then download the book so that you get your distinction material. Yeah, book. Yes. Those, if you have a sister for grade 10, grade 11, grade 12, we have distinction material for grade 10, distinction material for grade 11, distinction material for grade 12. So meaning that we have all the books which can make you succeed. Yes. You don't need even to stress if you are having this book. There is no question they're going to ask you in this life science, and this book cannot answer it. And the answer is already highlighted there. Yes. All right. Let's continue. Describe how meiosis lead to variation. How does meiosis lead to variation? Can this question be answered by this book? How meiosis? When you talk about meiosis, we are looking at the causes of variation, but these causes are based on meiosis. Because if you come here and look at the causes of variation, yes, let me show you so that you know exactly what they are looking at here, because you don't just need to write each and everything. No. Variation, difference among organisms of the same species, causes of variation, crossing over, random arrangement of chromosomes during metaphase, random fertilization, random mating, mutation. Where is meiosis here? Random mating is not meiosis, although it causes variation. What is random mating? I explained it. If you don't have it, you just go to Thunder Educ, and then you type there meiosis Thunder Educ. It will bring you this videos, you'll be able, or request them from this number 0848 Yes, request them, request those links, they will send them to you. All right, they're saying variation, random mating is not variation, because here whatever you see, you don't want to say that I want a girl with a big bums, I want a girl with big breasts, I want a man who is tall, who is light skinned. No, that is not random mating. Random mating, whatever comes to your face, you marry. That's it. It's like a chicken. When it's chasing this one and then another one passes across, then it goes with that. That's a random mating. So it is not about meiosis. Random fertilization, remember you have over, you release over 300 million sperms for a single ejaculation. Those are how many sperms you release in the vagina of the female during sex, just in a single ejaculation. So it means that if you ejaculate more than once, it means that you produce over 500 million sperms. But which sperm is going to fertilize the ovum? Check here. Which sperm among these sperms is going to fertilize the ovum? So still, this is not meiosis. So don't talk about this when the question comes and saying that describe how meiosis lead to genetic variation. So this is not meiosis. Yes, so don't talk about random fertilization. Then we go to random arrangement of chromosome during metaphase. Where do you find metaphase? You find metaphase in meiosis. Therefore, that is the first point you can talk about. Crossing over. Where do you find crossing over? We find crossing over here. We say that is found in prophase 1. So basically, you describe how crossover or crossing over led to variation, and also how random arrangement of chromosomes lead to variation. That's why once you explain that, you'll be able to get the marks. Mutation can occur during meiosis also, because here we have what called a non-disjunction. When does it occur? It occurs in anaphase. It can be either anaphase 1 or anaphase 2. So the non-disjunction is as a result, or this is the chromosome aberration or chromosome mutation. The whole chromosome has changed. The number have changed. So they can lead to variation, yes? So you can talk about it. But the major ones are, number 1, crossing over. When homologous chromosome form pairs during prophase 1 of meiosis, crossover occurs. And this leads to new combination of, when you have new combination of maternal and paternal genetic material in each cell. You see, they have created new genetic combination. And if new genetic combination has taken place as you see them here, before and then after, then it means that the organisms are going to be different. I always ask a question that don't take it personal. Please don't take it personal. Are you sure your father is your father? Definitely your mother is your mother, you know. Yes, but are you sure? Do you look like your father? I'm just asking a question. Yes, your father can be your father. But why don't you look like him? Why do you look like your neighbor? I'm just asking. Okay, yes. Why am I saying that? You have ever gone somewhere and they say that, when you look like someone I know, when you look like someone I know. Ne? But when that person has never seen you even a single minute, that is the first time, but he's resembling you with someone else. Why? Anyway, all of us, we came from Adam. All right. Meaning that we have some commonalities. Ne? Yeah. But when you go to science, I don't think they believe we came from Adam. All right. Depends on where you believe in or what you believe in. But yes, your father can be your father, but you don't resemble him because of what happened, the genetic variation which happened here during the crossing over. And then what happened? And then you ended up picking chromosomes from your mother, or you ended up picking chromosomes your mother maybe had from your grannies, or your father had from your grannies. So you end up not resembling your father. Even a single feature on you doesn't resemble your father. But doesn't mean that your father, that doesn't mean that what you call your father is not your father. You understand? Yes. So I'm just sometimes I ask you that question just to open your mind, asking you, so that you know that variation occurs, and variation is important because it brings about survival. Yes, survival. If you are exactly the same as your father or exactly the same as your mother, take a picture and you see. Do you want to be like your father, or you want to be better than your father? You want to be better than your mother? I don't think we work to be like a person exactly. We work to be more than that person you want. Yeah, if I can become better than him, not in a bad way, yes. Don't say that I want to be like Saidi, like me. No, don't even say that. If you are reading, you say that I want to be more than this. Say that I want to be more than him. I'll be happy. All right, but in a good way, yes. So how does meiosis lead to genetic variation? So what you need to describe is this crossing over. Talk about when does it occur? When does it occur? Yes. When you say when does it occur? Uh-huh. Someone is asking me I should send the link and stuff. Unfortunately, it can't allow now. Okay. Don't follow it now. Ne? You'll follow it afterwards. Ne? Yes. It can't. It can't. It can't now. Okay. I'll send it after the lesson. Ne? Let me see. Okay, yeah, I think. But after, please follow it after the lesson. Yes, I've sent it. We are saying that how? How? Number two, you talk about random arrangement of chromosome during metaphase. I'm gonna give you an example here. Which example am I going to use? How this random arrangement leads to different combination of things. I don't think when you go to class, your class, you sit exactly the same way you are sitting in a previous lesson. Unless you are so stingy to your table. Yeah. But if you're not, you first come, first serve. That's it. Or you go to the taxi, you are the same people who came, the transport you are using, the same people who were there before. In the morning, they are going to be the same people who were there, who are going to be there in evening or in afternoon. Yes. Do you sit in the same place? No, but it's the same people. If you start adding them, like being in pairs, you see that you have different combination. The combination, the person you are pairing in the morning is not the same person you are pairing in the afternoon. So that's how this random arrangement. We don't know either this chromosome is going to be this side or this chromosome is going to be this side. We don't know. We don't know which side this chromosome is going to be. Whether it's going to be on this side or it's going to be on the other side. Yes. So basically, that is this. Let's go back to the another question. So what you need to read, talk about, you talk about random arrangement of chromosomes. Yes. Sorry. You need to talk about random arrangement of chromosomes during metaphase, and then you also talk about crossing over. Those are the two things which you can talk about, and then you can also talk about mutation. But the mutation you are supposed to talk about here, it must be mutation which, because this is gene mutation, you see, this is gene mutation. Only if you are talking about the non-disjunction which occurs in meiosis. All right, let's go to another question. Another question is about karyotype. Ne? Another question is about karyotype. Guys, just 1 second. I think this is not something happened. I don't know. Check and see. Just a moment, because it's not detecting. All right. As it is, we are continuing. Let me ask the technician, to help me out. Mm. All right. They're saying that now, if I give you a simple task, né? Here, what is this? What is this? Tell me the answer. How do we call it? Individual 1 and individual 2. Which one is a male? Send the answers. How do you call this diagram? Yes. And then send me the answers here on your description. How do you call this diagram? And then which one is a male and which one is a female? Send me, and then I see, so that I can yes, talk about it. I'm waiting for the answer. Yes, I'm waiting for the answer. Who is the first person to answer? Who is the first person to answer? Where are you? I'm waiting. Which individual is one? Sorry, which individual is a male? Which individual is a female? And then how do we call this diagram? Just tell me the answer you know. Just the answer you know. Which one is individual? I'm waiting. I don't see you. I need to see you. Are you there? Lawrence, where are you? Okay. Femelo, where are you? Where's the answer? Which individual is male? Which individual is a female? And then how do you call this diagram? Someone is... Yes, how do you call this diagram? Okay. Femelo is saying that individual 1 is a male and 2 is a female. Good. Yes. Individual 1 is a male. Why? Because individual 1 is having X and Y. Yes? Yes, having X and Y. How do I know that this is X and Y? This one is long and this one is short. Well, this one is long and this one is long. Therefore, remember, we know that 1 is... Let me show you here. X and Y. X is longer than, bigger than Y. Therefore, if the 23rd pair is having the same size of chromosome, then we shall say that is a female. And if it has different sizes of chromosomes, we're going to say that is a male. Why? Because X is, remember, XX is a female. XY is a what? Is a male. So this is a female, this is a female, and then this is a male. Yes, thank you, Femelo, ne? Femelo. Yes, Femelo, send me a message on 0848 331738. Yes, I'll send you a book. Yeah, thank you very much. Yeah, so that you get a distinction. Thank you very much for your what? Your contribution. All right. They are saying that identify the type of chromosome. Okay, how do we call this? We call it a karyotype. Karyotype. When you say the word karyo, in science, it means a nucleus. So when you say karyotype, which type of nucleus do you have? Which type of nucleus do you have? How are these arranged? Yes. So we are saying that, thank you very much. this is a karyotype. This is a karyotype for a female, and this is a karyotype for a male. This is a karyotype for a male because X and Y, and this is the karyotype for female because of X and X. So identify the chromosome numbered 1 to 222. Look, this booklet is indicating this exactly because there is a 2019 they brought this question of description of a karyotype. Human cells are made up of 46 chromosomes arranged in pairs. Each pair is different from each other by shape, size, and genetic composition. From pair number 1 to pair number 22 is called orosome. There is no question you're gonna be asked in life science anywhere and can't be answered by this distinction material. Tell your teacher. Can think about any question which is supposed to be examination guideline and then can't be answered by this question. Okay, let's see. Identify the chromosome number 1 to 22. Here, I mentioned it very well in this book. Say that 1 to 22 is called orosome. So the answer is orosomes. We call those chromosomes orosomes. Question 2. They're saying that each pair shown in each of these pair shown is a homologous pair of chromosome. State the origin of each chromosome in homologous pair during the zygote formation. Where do they come from? This book said it. It said it. We said it here. We said it here. Here. Yeah. Not here. Here. Yeah. One chromosome will come from a sperm. One chromosome will come from the ovum. And then you form a zygote. A sperm does not come from a lady. It comes from a female. So this is coming from the maternal line. And then it comes from the paternal line, and then this comes from the maternal line, and then you form a zygote. That's what they're asking you here. They say state the origin of each chromosome in the homologous pair during the zygote formation. Where do they come from? From the sperm of the father, and then another one, and another one from the ovum of the mother. I think I'm clear. List 3 characteristics of chromosomes in homologous pair have in common. List 3. I'm not the one who said this paper. No, this is the national exams. I mentioned it here. If you read this book, you will find the answers what you need. Describe, how do we call this? How do we call this? What do they have in common? Here is the answer. I mentioned it here. Say that these are pairs formed by shape. They have the same shape. That's number 1. They have the same size. That's number 2. And they have the same genetic composition. This is in terms of locus, the position where these genes are found. Yes, that is basically what we need from you. The same genetic composition. Same genetic composition. So, where are they found? The locus, the position, because locus means the position of a chromosome. If there are many, we call them loci or loci. I don't know whether they call it loci or loci. Yes, loci. So the same genetic composition. I think I'm clear there. I'm clear there. Okay, now let's go to the next one. The next question. Explain one observable. What you see, not what you studied or functional. No. When they ask you observable, they are looking for the structure, features. One observable reason why 2 individuals are not identical twins. Why are they not identical twins? Why are these? Guys, don't forget that identical twins come from one egg. This egg, it has a nucleus, remember? Nucleus, jelly-like layer, and then the nucleus, and then the sperm. Why are these karyotypes are known for identical twins? This egg must be fertilized by one sperm. By one sperm. So this sperm can either be X or it can be Y. So if it is X, then automatically this sperm must split this egg into two. So that now you have two individual one and individual two. So if it was X, they are going to be girls. If it was Y, they're going to be boys. So now, there is no identical twin. We don't have any identical twins in this world, and these identical twins are of different sex because two sperms cannot fertilize one ovum. No, impossible. The same way one sperm can't fertilize two ovum, then two sperms cannot fertilize one ovum. Yes. So, because this is male and this is a female, therefore they can't be identical twins. Give one observable reason. It's because chromosome pair 23 has X and Y in individual 1 and X and X in individual 2. Yes, that is the perfect answer you can give. So why? Because chromosomes cannot, individuals cannot be of the same sex. Cannot be of the same sex. So now this is another karyotype. Yes, still is a human karyotype. Is it a human karyotype? We shall see. Yes, because of the 23 pairs of chromosomes. But there is something which we are hiding there. We're going to see. Let's see. Name the disorder represent. Where is the disorder? In most cases, when they talk about disorder, we are talking about we are talking about chromosome number 21. That's what we are looking at. Chromosome number 21. So look at chromosome number 21. What is happening? Eh? What is happening there in chromosome number 21? You see what is happening? How do we call that? How do you call that? Yes? Is there anyone who knows how do we call that? Who knows how do we call that? Okay. This is Down syndrome. Down syndrome occurs on chromosome number 21 as a result of failure of the chromosomes to separate. When the chromosomes fail to separate during metaphase, then we call it Down's syndrome. During anaphase, we call it Down's syndrome. How do we explain it? I'm supposed to explain for you, but let me just show you how this Down syndrome is being brought about. There we go. Non-disjunction is here. Non-disjunction. Okay, let's see. What are they saying? Name the phase in meiosis which when the non-disjunction occur. We said it here. Non-disjunction is the failure of the chromosome to separate or chromatid to separate during anaphase. So it occurs during non-disjunction occurs during anaphase. Then they are saying that name the type of mutation that result of non-disjunction. Remember I said that this kind of mutation, because we are changing the number of chromosome, it can't be gene mutation. It is chromosomal mutation. It's the whole chromosome which is changing. We can call it chromosome mutation or chromosomal aberration. Why do you go for aberration? Stressing you. Chromosome mutation. Simple. Simplify your life by taking. Some kids, they want to write these big terms that they scare the examiner. There is no way you're going to scare the examiner. You are just scaring yourself. Because the examiner, we already know. By the time we set that question, we know even the alternatives you might say. That's why sometimes we put them there in the memorandum or in the marking guideline. Yes. So there is nothing you're going to scare the examiner that by writing big terms. You are just stressing yourself, and those big terms can lead to wrong spellings and then leads to loss of marks. So simplify your life. Don't think that when you are unique or you are giving different answers than you like from others, then you get more marks. You get the same mark like the other person who has written a simple answer. You get. So name the type of mutation that is as a result of this non-disjunction. This non-disjunction of chromosome number 21. The answer is chromosomal aberration or chromosomal mutation. So choose what you want to go with, because it's the same thing. Diagram below represents the distribution of chromosome pair 21 as it appears in each gamete. So non-disjunction, yes, non-disjunction. Let me just show you how it operates. This is how non-disjunction occurs. You have chromosome. This is a cell. Let me just say chromosome. You have this cell has 4 chromosomes. I'm describing non-disjunction. Non-disjunction. So what happens here in that non-disjunction? How does it lead to Down syndrome, basically? Yes, because this is a little bit challenging to students, and then they end up losing marks. So this chromosome is supposed to separate this side to go this side, and this one goes this side. But during non-disjunction, there is failure of these chromosomes to separate. All set of this chromosome goes this side. So what happens? So I'm gonna have this cell, 2 cells. You see? Now this cell is gonna have this and this. Ne? So they have moved to this side. Ne? Okay, let me draw it here so that you see it clearly. So this cell, now all of it has moved to this side. So I'm going to have 1, 2, plus, let me change the color. Let me change the color, plus this one, which is 3. You see? And then now this side, remember this and this, they were supposed to separate. They separated it correctly. Yes? But this and this, all of it came this side. I expected to have this side, but it didn't go there. Failure of the chromosome to separate during anaphase. Therefore, now this cell is going to have, is supposed to have this one from this side, and also this one must have one from this side. And also, did I change? Yes. Good. This one, yes. Okay, yeah. It's going to be this and this. So it's supposed to be 2 here, 2 here. Unfortunately, I have 1, I have 3. So I have 3 instead of 2, I have 1 instead of 2. So this is, it has short of 1 chromosome and then extra 1 chromosome. You understand? Now, if this 1 chromosome, this non-disjunction, if this 1 chromosome fails to separate, and then now it fertilizes the normal ovum. For example, now this fertilizes this. If this is a human cell, now let's state that this is a human cell. Remember, I told you that they only talk about human cell, and human cell has supposed to have 46 chromosomes. So in meaning that after meiosis is supposed to be 23. Now this one is supposed to be 23, but it's not 23 now. It's 24 because it has 1 extra chromosome, and this one has 22. So now it's gonna be 24 plus 23. They're gonna be 47, and then this one 22. No. Yeah, 22 plus 23 is going to be 45. If this non-disjunction occurs on chromosome number 21, then it becomes what? It becomes trisomy 21. And this condition is what you call non-disjunction. This is what I said. Each nucleus should have 23 chromosomes after meiosis. If non-disjunction occurs, one nucleus will contain 22. That's what I said. And then the other one will have 24. That's what I said. When either of these resulting gamete fuse with the normal gamete, it means that 22 with 23 is going to be 45. 24 plus 23 is going to be 47. It's either going to be 45 or 47. You understand? This is what I'm saying. This leads to non-Down syndrome. But this non-disjunction, it must be on chromosome number 21. Because if it occurs on another chromosome number, it won't lead to non-disjunction. No. It won't lead to Down syndrome. It will be another syndrome. Yes. If it occurs on sex chromosome, it can be Klinefelter syndrome, Turner syndrome. So it depends on what chromosome number is it. Yes. So women who are under the age of 40 have a great chance of having this Down syndrome. Yes. So it means that when you study, you get your university, start working, get married, have children before 40 years. Yes, so that at least you have less chances of having this condition. I'm not saying that if you're 40 years, you have a child of Down syndrome. We're saying that less chances if you have kids who you have kids when you are still less than 4 years. I'm talking about ladies. Men are always fresh. Anytime. Even if I'm 70 years, I'm fresh. I can have proper kids. Yes. And men, they don't stop. Ladies, yes, for them they stop, but men, they don't stop. Anyway, let's go to the next question. Diagram below represent distribution of chromosome pair 21 as it appears in gamete at the end of meiosis. Let me know if you have any question. You have any challenge? Please let me know. Ne? Yeah, I'll answer your question. This is a live thing. Ne? So let me know so that I can crush that question now. Because I say that by the end of this, you must be able to answer all questions concerning about meiosis. Yes, you must. Yes, all questions concerning about meiosis, you must be able to answer them. Explain why the gamete represented by C and D do not have any chromosomes. So what happened? Non-disjunction occurred. When did they occur? It occurred in when? In anaphase. So it means that explaining why gametes C and D don't have chromosomes. It means that non-disjunction occurred in anaphase. Check what happened. If it was anaphase 2, yes, only one would have been without. This one would have been with the 1, and then another one with the 1, then 2. But now it happened in anaphase 1. It happened in anaphase 1. Yes, it means that this chromosome started with, let me show you how it led, how did it come here? Yes. So if this is the cell, né? The cell had 2 chromosomes. Wabona? It had 2 chromosomes. How did this came about? It had 2 chromosomes. And then, no, I'm saying 2, not this one. Sorry. Let me do it again here. This is the cell. Yes, it had 2 chromosomes. If this is metaphase, yes. If this is metaphase, yeah. Now it's supposed to split during anaphase. Ne? This one must go this side. This one must go this side. We are explaining how did it come. So what happened? All of it went to one side. For example, it has gone this side. Ne? So what happened? This one has 2 of them, 2 chromosomes. And then this one has nothing. You see? This one has nothing because all of it has gone to the other side. So this one is empty. So now meiosis 2 occurs. When meiosis 2 occurs, what's going to happen? When meiosis 2 occurs, this one is going to split. It's going to split like this, whereby now this one is going to have one chromosome from this side and one chromosome from this side. And then this one is going to have one chromosome from this side and one chromosome from that side. What about this one? When it undergoes meiosis 2, because it's empty, it's going to be empty. And another one also is going to be empty. Hence, it brought out this. So it means that this occurred in anaphase 1. Sorry. In anaphase what? In anaphase 1. You see? So you have to know these techniques. Yeah. In anaphase 1. If it happened in anaphase 2, still you will have seen it only one cell would have been without. If the gamete A, where is A? Is involved in fertilization, describe how this will lead to Down syndrome. So if it happened on, did they say it? Yes. You see? And then they say it is a human. Then automatically, now we know. Now we know it occurred on chromosome number 21. It means that if this one has gamete A, it has 24. If it fertilizes the normal gamete, which is 23, the ovum, so the individual is going to have 47. Actually, this is what they are showing you here. This. It's going to have an extra, hence Down syndrome. Due to the process of crossing over, chromosome in diagram 1, A and B appear different to each other. Identify the phase of meiosis during which crossing over occurs. You see? When does crossing over occur? We say that it occurs in prophase. Prophase. Which prophase? Don't just say prophase. If you say prophase, we cross. You know, crossing is nicer than giving it. It's very difficult to give you a tick than giving a cross. Yeah. Yes. When you have many questions, correct. The hand keeps on doing this. Correct. Is when you have wrong, you end up having even the correct answer is wrong. So please try to read, so that this thing doesn't happen. Yeah. Yes. They're saying that describe how the event. They're saying that identify the phase. This is it. Prophase 1. This is what I said here in the booklet. Crossing over. Yeah. Process of crossing over. Yes. Process of crossing over. Yes. I said it here. And I said it here also, the real phase of crossing over. This homologous chromosome line up together. One chromatid of each chromosome overlap with the chromatids of this homologous pattern, and then the crossing over takes place at a point called, wherever you see this is a marking point. Yes. Just download the book. Use this book. Make sure that you use this book. I guarantee you a distinction. Yes, last time I was showing you the kids. A teacher was very happy, sending me a message that, sir, I'm thanking you for using your booklet. I managed to get 12 distinctions. 12 distinctions. I salute you. I salute that teacher, and thank you very much. Keep your work up. You want a distinction? Yes, that book. All right. Let's continue. Diagram below, we are almost done. Diagram below represent all the chromosomes in a cell that is undergoing normal cell division. Cool. Type of cell division is resulting in the cell, in the diagram. Which kind of cell division is this? It's meiosis. Meiosis. So the answer is meiosis. The phase of the cell division during which the chromosome behave as in the diagram. How do we call this? This phase. Is it prophase? Prophase. Prophase. If you say prophase just like that, I said we cross. Prophase 1. Then that where in the cell, in human cell, female would the type of cell division occur? I said it very well. I've been showing you over and over. I told you that there is no question in this. They're gonna bring, and it can't be answered by this booklet. I'm telling you. You want distinction here. So the answer is female ovary. They're saying that where give the letter and the name of the structure that has that attaches to spindle fibers. Spindle fibers. We talked about it. This C and is centromere. There's some students who write this spelling very wrong. Centromic, centro. They have the way how they see centromere, centromere body, the central body. That's what it means. They're saying that give the, we are done. We are saying that which letter is C? The answer is C. And then the name is centromere. How many chromosomes will be found in each daughter cell at end of this cell division? How many chromosomes will be found in daughter cell? Did they say that this is a human cell? Let's go back and see. Undergoing normal. They didn't say that this is a human cell. So I cannot think about human cells here. How many chromosomes? I can't. So I just have to go back and see. It's 1. So they're saying how many chromosomes will be found in each daughter cell? Yes, at the end of they are not talking about human cell. How many chromosomes will be found in each daughter cell at the end of this cell division? Meaning that at the end of meiosis 2. Meiosis 2, so the answer is going to be so it's going to split. So the answer is going to be 1. It's going to be 1. And they are saying that okay, they didn't talk about human cell. Study the diagram below. Okay, what is A? A is a chromosome. B is a chromosome. Both of them homologous chromosome, but because they're undergoing crossing over, we call them bivalent or bivalent. Bivalent. That's the terminology we use. And then this is the crossing over chiasma, chiasma or chiasma or chiasma. This is centromere, centromere, and then this is chromosome, just normal chromosome, and then this is chromatid. So I think we have identified all the parts. Name 2 ways in which structure A and B are similar to one another. A and B. We talked about this. It's the second time we are talking about it. Yes, we talked about it. That this is you just look at it here. At karyotype here. Same shape, same size, same locus of the genes. Yes. Same shape, same size, and same locus or loci of the genes. They say describe the process of at D. Is at D. They're looking for crossing over. I also talked about it. Homologous chromosomes align together. One chromatid, you see,
these chromosomes, they align together. One chromatid of each chromosome overlap at with the chromatid of its homologous pair, pattern or pair at a point called chiasma. So you see, they overlap at this point called. So this is crossing over. This is that is also another marking point. They're saying that chromosomes separate in such that each has new genetic material. So they separate. Once they separate, you see that they have new genetic material. It's also a marking point. Yes. So basically, that's what they are looking for. So these marks, these 5 marks, they come from here. 1, 2, 3, 4, 5. Done. So follow that book. For that book, you'll be able to get a distinction. You'll be able to get a distinction. Then they are saying that, yes, we are saying that certain species has 42 chromosomes in its muscle. When you say muscle, we are talking about body cells, diploid, 2N. Yes, body cells. Give the number of chromosomes in each sperm. So it means that sperm is always having haploid. So it's going to be 21. And then skin cell is the same thing with the muscle. So it's going to be 42. They are not saying that this is a human cell. They're saying it's certain species. That's why you should not only talk about, yes, ova of its female counterpart. It's the same thing like the sperm. So it's going to be 20, 21. Describe how non-disjunctional lead to Down syndrome. 5 marks. I've explained this and I've shown you how to explain it. And I've shown you that when you use this book, nothing can stand in front of you. Here is the mark. Each nucleus should contain 23 chromosomes after after meiosis, but if non-disjunction occur, 1 nucleus will contain. So I've explained this several times today. So meaning that basically, in other words, there is no way you're gonna have a question and that question cannot be answered. There is no single way. There is no single day. There is no way how you're gonna get this question, and this question cannot be answered by this booklet. It's Saidi. There's a student who asked me a question that about the DNA base calculation. So let me just go through that base calculation. After that, we will finish up with the terminologies, the base calculation. We're gonna finish up then with the these terminologies, and then by the end of that, then you'll be able to answer anything related to this. Thank you very much. Get a distinction. Let's go straight to the booklet. The question is here. I tried to do questions about base calculation, and I left this question, but many students are asking me about how do we calculate this question. But this question is supposed to be your practice, and you come back to us, and then we see if you understood what we meant. Anyway, let's not waste time. If DNA has 5,000 bases, only 500 is guanine. What is the ratio of thymine to cytosine? It means that if you have 500 bases, 5,000 bases, and this is the total for DNA. Ne? Only 500, and if you check here, they are saying that only 500 is guanine. Only 500
It means that this is equal to guanine. Now, what are they looking for? They are looking for thymine to cytosine. They are looking for the ratio of thymine to cytosine. So if use the other method, which I call the Bible of DNA, remember that adenine goes with thymine, and then guanine goes with cytosine. So if you have guanine goes with cytosine, and guanine is 500, guanine is 500, definitely cytosine is going to be 500 because these are complementary bases. If you have 10 left legs, definitely you have 10 right legs. If you have 500 guanine, because they go together, definitely you're going to have 500 cytosine. So when you combine the two, you're going to have 1000. But the total number of bases in the DNA, these ones, all of them is 5,000. Is 5,000. So what does it mean? It means that if I remove guanine cytosine, as this is equal to 1,000, yes, the balance will be for this. So I'll come here and I say 5,000 minus 1,000. It will give me how much? 4,000. But this 4,000 is for both adenine and guanine. And adenine to guanine, they are equal. Therefore, I have to divide it by 2, and then I get 2,000. So it means that adenine is 2,000, and the thymine, adenine is 2,000, and the thymine is 2,000. So what does it mean? It means that um, if I add this, and this, I must get 5,000 because guanine is 500, cytosine is 500, adenine is 2,000, and thymine is 2,000. I must make a total of 5,000. But the question is looking for the ratio of thymine to cytosine. Therefore, thymine is 2,000, and then don't change the arrangement cytosine to thymine. They say the thymine to cytosine. And then cytosine is 500. So I divide so that I get the simplest ratio. For example, if I divide by 500, I'm going to get 1. And if I divide by 500, I'm going to get 4. Why do I divide by 500? Because it's the simplest here. When I divide by 500, and 500 goes in 2,000. So that now I get the ratio of 4 to 1. There are sometimes you're going to get a ratio of, for example, 4 to 2. Ne? For example, uh, um, for example, I can give you an example of 2,000. Yes, I'm just imagining to 1,000. Ne? For example, if you divide by 500, you're going to get 4 here, and you're going to get 2 here. This answer is not correct. Is wrong. Why? Because this is not the simplest. So the ratio must be the simplest. This answer can be divided more to get the lowest figure. How? If I divide by 2 here, I divide by 2 here, I'm going to get 2 here, and I'm going to get 1. You see that this one is simpler than 4 to 2. So when they ask you the ratio, it must be the simplest version. And if you divide it, it must not be in fractions. It must be a whole number, and it must be the simplest in such a way that there is no way you can divide and you get a whole number. So that's what you call the ratio. So in this case, the answer is going to be 4:1. And they can confuse you in objective. They say A, B, C, D. And the first answer here is 1 to 4. Because when you have seen 1 and you have seen 4 somewhere, you just circle or you just say that that question is A. It's going to be wrong. You have to take note and care before you try to answer these questions. Yeah, then you'll be able to get the distinction. Don't rush. If they give you the paper for 2.5 hours, examiners are not, let me quote it. I'm just quoting it. They're not stupid to give you 2.5 hours. Why do you do the paper in 1 hour? You think they are stupid to give you, I'm quoting it, yeah, don't take it that way. Trying to show you that use all your time. Don't sleep in exam. Why should you? When you sleep in your exam, you are sleeping in your life. You are sleeping in your future. So please use all the time you have. Don't rush. Read the question more than 2, 3, 4 times. Don't pressurize yourself. Yes. Try to go slow. Make sure that you manage your time. Remember at the first I said that as long as you know what you call time management, don't forget that exam is also time. If you don't finish on time, you won't be able to get a distinction because you won't know what is in your head. Anyway, let's go to meiosis. Meiosis, what is meiosis basically? We say that meiosis, as a simple recall, we say that meiosis basically is a type of cell division whereby you have one cell divide into 2 cells, and then 2 cells divide into 4 cells. And these cells are half the chromosome number of the original cell. For example, if the cell starts with a 10, definitely it's going to be 5, and it's going to be 5 chromosomes. You see, it's not a must that it's going to be, the question for meiosis is going to be from human cell. No, it's not human cell always. If it is a human cell, they will mention it in the question that this is a human cell. Then now you can think about the 46 chromosome. But if they don't mention about human cell, don't think about that this is a human cell. It must be what? It must be based on what you see. If you see a cell with the 5 chromosomes or 3 chromosomes or 6 chromosomes, it means that those are the chromosomes. Why do you set your own question and you call these 46? What do I see here? I see 6 chromosomes. Yes. Don't start to imagine that all the questions in meiosis is supposed to be human cell. No, because the topic is not about human meiosis. It's about meiosis, whereby it can be any kind of. These are some of the things which make students to confuse meiosis, and when it comes to how many chromosomes are found. Anyway, we're going to see such questions and you see how we can deal with them so that by the time you leave this lesson, at least you are perfect with meiosis and there is no question it can come and you fail. Ne? Yes. Let's go to the first question. Let's start with some of the terminologies in meiosis. Chromosomes that are non-sex chromosomes, there are 22 in pairs. There are 22 in pairs. You understand? There are 22 pairs of chromosomes in diploid cell. So how do we call those chromosomes? There are 22 pairs of chromosomes in the diploid cell. So how do you call this? This is a small description to show you a hint. What is it? And based on this question, They have given you the answer. You understand? Yes. So the chromosomes, remember that you have two kinds of categories of chromosomes in a human cell. You see, they said it in a human cell. They said it. Whenever they are talking about human, they will talk about it. So you have from pair number 1 to pair number 22. We call it orosomes. Call it orosomes. Mind about the spelling. And then the 23rd pair, remember these are 2, ne? So we call them gonosomes. See? Yes. So what are they asking here? They're asking for, yes, chromosomes that are not sex chromosomes. So the answer, is it autosomes or gonosomes? The 23rd pair, remember it's made up of X, is made up of what? X and Y. X and Y. Yes. So they are looking for this. So the answer is autosomes. So the answer here is going to be autosomes. Autosomes. Yeah. Organelle in cell in cytoplasm of the cell, which give rise to spindle fibers during meiosis and mitosis. So here they are looking for the organelle, basically in animal cell. So the answer is going to be centrioles. But because they're looking for the organelle, so it's going to be centriole. If they're saying organelles, it's going to be centrioles. Yes. Structure that hold the chromatids together to form the chromosome, form a chromosome. How do you call that structure? They are looking for, if this is the chromosome, how do you call this structure? Go slow, you would know that meiosis is nothing. You have to get a distinction, and not just a distinction. We are talking about, you will get a good distinction, 95% and above. So centromere, centromere, centromere. Please repeat after me. Centromere, centromere, centromere. Because I'm saying this because when it's time for marking, you see that students try to write words which you have never heard about. It's because you don't repeat these words to stick in your head. Centromere. Yes. Point where crossing over takes place between the chromatids of the homologous chromosomes. This is where the exchange of genetic material is happening in prophase 1. So here they are showing you, or they are saying that you have a chromosome and another chromosome. So you have this point and you have this point. Yeah. Okay, let me draw it nicely. It can happen on top or it can happen on bottom. See now, it can be this or it can be this. On. Yes. I see. So they are looking for this point. They are looking for this point where two chromatids of homologous chromosome cross over. How do you call that point? We call it chiasma. Yes. So the answer here is gonna be chiasma. It's gonna be chiasma. Sorry. Yeah. It's gonna be chiasma. How do you write chiasma? How do you write chiasma? Student, how do you write it? Okay, is CHASMA.
IA chiasma. 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 If there are many, we call them chiasmata with the TA. But they're saying a point. They're not saying points. So if it's a point, chiasma. Ne? If there are many, so this is out. If there are many, chiasmata. Yes. They're saying it is a single thread of a double-stranded chromosome. How do we call that? Because if it is a chromosome, ne? Look, if it is a chromosome, ne? This is a chromosome. How do you call this part? That's what they are looking for. Chroma. How do you call it? Chroma. Chromatid. Is a chromatid. This is I. Chromatid. So the answer here becomes chromatid. I should take note of that. Yes. Is a chromatid. Structure made up of two chromatids joined by the centromere that carries the hereditary characteristics within the DNA. How do you call those structures? Remember, we said that in interphase, that structure duplicates. And in prophase 1, it condenses. And then now you can see that structure when you're under a microscope. It becomes visible. How do you call that? Structure, which is visible, which divides during meiosis. So that structure is called the chromosome. They're saying a structure, they're not saying chromosomes, not saying structures. Yeah, you should take note of that. Ne? Yeah, a scientist must be always observant. Then they're saying that complete chromosomal number represented in pairs, which is characteristic of an organism, complete set. You either have half of it after meiosis, or you're going to have a full set. So a full set, which number is that? We call it diploid number. Diploid number. Yeah, sometimes we call it 2n. And then the other way, we call it haploid, which is 1n. Ne? So this one is a diploid. This refers to each chromatid after it splits from its sister chromatids during anaphase 2 and is moving towards the poles. How do you call it? They split. But they are saying during anaphase, they're saying during anaphase 2. If it is during anaphase 2, so what does it mean? What does it mean? It means so if you check on the process of anaphase 2, that phase, you will see that you have a cell, for example, you have chromosome and you have a chromosome. Remember, at this point, it's just a chromosome. Yeah. If this is metaphase 2, so what happens in anaphase 2? This chromosome is supposed to split so that you have one part here, have another part. It's moving this side. It's also moving this side. And you have another part here. It's moving that side. You have another part here. It's moving that side. If this is the pole, so this is what they are looking for. So they said that
refers to each chromatid after splitting from the sister chromosome. How do we call that? And is what is it referring to that? To the sister chromatid after splitting from each sister chromatid during anaphase 2 and is moving towards the opposite poles. So basically, it is moving, once it reaches the poles, they are chromatids now, but once they reach the poles, how do we call them? They are daughter, yes, they are daughter chromosomes. They are daughter chromosomes. So when you come back here, the answer is going to be daughter chromosomes. So that is the answer. What is going to be in that regard. And then they're saying, so that's why daughter chromosomes refers to each chromatid after it splits from its sister chromatid during anaphase 2 and is moving towards the poles. See, now it makes sense. Makes sense, ne? Yeah. All right. They're saying that haploid cells, remember, we said haploid, those are cells which have reduced the number by half of the normal cell. If the normal cell is having 10 chromosomes, the haploid are going to be 5. The normal cell has 4 chromosomes, the haploid is going to be 2. If the normal cell has 46 chromosomes in terms of human, the haploid is going to be 23. So they're saying haploid cells which contain half the chromosome number of the diploid generation. They give you example of the cells are over sperms. They're trying to give you an example. Yes. So basically, when the question comes, it comes like this and it stops there. So how do you call these cells? We call them gametes. So when you say gametes, it means that we are talking about ova and sperms. These are gametes, né? See? And then you also have in plants. Yes, because they are not saying it is in human. Yes. So gametes are haploid cells which contain half the chromosome number of the diploid generation. See, makes much more better sense. And they're saying that the unit of heredity transmitted in the chromosome, which controls the development of the characteristics. How do you call that unit which is being transmitted in the chromosome or along the DNA? Just a section of DNA that controls a particular characteristic. That's what they're trying to say here. Oh, that's what they're trying to mean. Just a section, part, portion of DNA. Remember, DNA, chromosome, genes. I've said the answer. Okay, there. This section is the same thing but different scales, as I explained it before. So the answer is gene. Gene. Sex chromosomes. How do you call sex chromosomes? Just saying that is enough. Sex chromosomes basically either is X and X or X and Y. So we call them gonosomes. Remember, I said it here when I was trying to describe it that you have 1 to 22 is called autosomes. The 23rd pair is called gonosomes, and they exist as X and Y. You see? Yes, that's what they're looking for here, gonosomes. Half the chromosome number of the chromosome present in the gamete after meiosis has occurred. Half the chromosome, you see? 2N, 1N. Therefore, I said that this is called haploid. Haploid. Yes, haploid. And then they're saying that They're saying that maternal and paternal chromosome having the same shape, size, which are paired but different or differ in the genetic material. How do we call that? Paternal and maternal chromosomes, it means that one from the mother, one from the father, but they have the same shape, same size, meaning that the location of the genes along these chromosomes is the same, but they differ in genetic material. What do they contain? They differ. It's like having a complex with the same shape of the houses, same gate, everything is the same. But when you enter inside the house, what is inside is different. Same shape, same design, same color, everything is the same. But when you enter inside, you'll find something different. Same structure of chromosome, same size, same, they move together, same. This is from the mother, this is from the father. How do you call this? Homologous chromosomes. Yes, basically, that's it. So, when they ask you that, so the answer becomes homologous chromosomes. Yes, homologous chromosomes. Pair of homologous chromosomes that chromosomes physically held together by at least one DNA crossover. How do we call that? How do you call those chromosomes during crossing over? I said it and I said it and I'm going to say it again. It's called bivalent or bivalent or bivalent, depending on where you're coming from. Mother, make sure that the spelling is correct. From the mother of female parent, how do you call it? Paternal. They said even it here. So this is the answer. Yes, paternal. Did I say paternal? Maternal. Yes, because it's from the mother. Then from the father, paternal. So the answer becomes maternal. Yes, maternal or paternal. Sorry, or maternal. A process of cell division whereby the chromosome number is halved for the production of haploid gametes. What do you call that? Meiosis. Meiosis. Remember that when we ask you the functions of meiosis, what do you say? We say that production of haploid gametes. And these gametes include sperms and ovaries. Isn't it? Yes. Production of haploid gametes. Yes. If you check it here, is basically I've not been referring this to, yes, I've not been referring this to the ova, and these are gametes. Ne? Yes. So basically, you produce haploid gametes, and this could be ova or sperms. That is in plants. Sorry, in animals. And then in plants, so these are all the things we have been talking about: haploid, diploid, haploid, diploid. So you should not confuse yourself. This is what we have been talking about. Autosomes, 22 pairs, autosomes. One pair is gonosome. If you read this book, you will get a distinction. There is no question which is going to come and you cannot answer that question. You have, if you really have and love this book. Whether is the teacher or whether what? Yeah, this is the production of importance of meiosis. This is what I'm saying. Check the thing. Process of cell division whereby chromosome number is halved for production of haploid gametes, sperms and ova. Check here. They're saying production of haploid gametes. And then this is as a result of the halving effect of this meiosis. It means that it leads to dividing the chromosome number into halves. All right. Another question as we winding up with these terminologies. Process of cell division where resulting the cells have the same chromosome number as the original parent cell. How do we call this? Process of cell division whereby the resulting daughter cells have the same number. This is mitosis. So basically the major difference in mitosis and meiosis is mitosis. Meiosis, it starts with 2N and ends with 1N. Mitosis, you start with the 2N, you end with the 2N. Yes. Mitosis results in 2 cells, meiosis results in 4 cells, but all of them are. Mitosis is same genetic composition. Meiosis is different because of crossing over. Some from the parent, that is the father, some from the parent, that's the mother. Homologous chromosomes, yes, do not separate due to failure of the centromere to divide during meiosis 1 and 2, resulting in gametes which will have either an extra chromosome copy or another gamete will have less. How do you call it? How do we call that? And we talked about that in this failure of the chromosomes or chromatid to separate during anaphase, that is during meiosis 1 or meiosis 2. They're saying anaphase. They didn't say anaphase 1 or anaphase 2. Any anaphase can happen. And if you check here, is meiosis, it can be anaphase 1 or anaphase 2. So the answer is non-disjunction. Non-disjunction. So the answer here is going to be non-disjunction. Father or mother parent. This is called what? It's called, remember we said that the mother is called maternal. And then the father is called paternal. We talked about it here. Yes, we talked about it here. Paternal. So it's called the paternal or paternal. Paternal. Yes. Refers to the chromosome as it appears after DNA replication takes place. Hence, each replicate the chromosome is made up of 2 chromatids joined by the centromere. How do you call that? This refers to a chromosome as it appears after DNA replication. Remember, during telophase 2, so the chromosomes, remember, you form 4 cells, né? I'm going to draw 2, but these chromosomes are not double-stranded. They're just single-stranded. That's why sometimes we call them unreplicated chromosomes or single-stranded chromosomes. So what happens? Even this one, the same thing. So what happens? During interphase, this unreplicated chromosome must replicate. And the moment it replicates, what happens? It becomes double double-stranded chromosomes. So because it has become double-stranded, sometimes we call it replicated chromosome. I indicated it here in our introduction when I was introducing meiosis. Say that this is homologous chromosomes, but these are unreplicated chromosomes. That is before DNA replication. That's why I'm saying the unreplicated chromosomes. Or we can call them single-stranded chromosomes. But after DNA replication has occurred, then we call them replicated chromosomes or double-stranded chromosomes. Why? Because they have two strands. So if you say that double-stranded chromosome or replicated chromosomes, this refers to chromosomes as it appears after DNA replication takes place. Hence, each replicated chromosome is made up of two chromatids joined by the centromere. This is exactly what is trying to say before and then after. So this is the answer in that regard, which is replicated chromosome. So the answer becomes replicated chromosome. Replicated non-diploid body cell. Normal diploid body cell. How do you call those cells in the body? We call them soma. Soma means body. Somatic cells. So microtubules that form during cell division, which radiates out from the centrosome and draw the chromosome to opposite poles. How do you call them? We call those fibers spindle, spindle. This is the spindle fibers or spindle threads. Yes. They're saying that it refers to the chromosome as it appears before replication. The same thing which I've tried to show you here. Now they're saying before. So it becomes unreplicated chromosome. So it becomes what? Unreplicated chromosome. Then the morphological and physiological difference that can be seen between the members of the same species, like the difference among organisms of the same species. So we call it variation. Variation. And then lastly is the resulting in diploid cell after fertilization. When two haploid cells, that's this, the gametes, when they divide, they form what we call the zygote. So when they divide by meiosis, then become haploid, then they fuse, then they form diploid. As indicated here, I showed you here that two haploid, then they form a diploid, which is a zygote. Yeah, so that's what we're saying, that resulting in a diploid cell after fertilization has occurred here. Yeah, download this book. You won't regret. You will never regret. Summarized, don't forget that different color means a marking point. Yes. I think I've tried to answer questions concerning about the terminologies about meiosis. Our next class, our next step, just in a few minutes, is gonna be the going through of all these questions. I'm coming. I'm gonna explain everything which you see here, which you see. There are a lot of questions. So by the